Our next speaker is from Case Western Reserve University and is going to be talking about something a little bit more applied, Plasmodium.
Thank you. Thank you. And thank you for the opportunity to come here and see a lot of interesting work done on, among other things, malaria. Again, the focus of my presentation will be not exactly in the direction of applying it to specific models of control interventions and other things, but sort of conceptual understanding one thing, again, important thing, the role of immunity as a selective driving force for parasite diversity, multi-strain malaria. Okay, in general, again, this is a very general topic. It's not only malaria. It's not malaria only, but one knows many other examples where selection plays an important role, and then different factors can affect these things. For instance, drug resistance is a big issue, because as everyone knows, for microbial infections, malaria as well has many applications for control. Another thing also important, that's what I will focus on, the role of immunity, how immunity can shape, how parasite immune interaction can shape the evolution of parasite. And of course, examples that we know well from very recent, and not only recent, respiratory infections like COVID, flu, RSV, observed patterns, they show, and definitely what happens, they evolve. COVID does evolve, and one reason for COVID to evolve is to evade immunity. So again, immunity is important driving force here. Understanding how this mechanism works is, of course, going to have important implications for many different things, vaccine in particular, whatever control mitigation when tried. One question, and again, something that partly motivated my work in the context of malaria was asked by Andrew Reid back in 2008, and here's the following question: can imperfect vaccine drive evolution of virulence, malaria parasite? Now, evidence, just some, I won't comment too much on this. And again, it's not so much to address this question by Andrew Reid, I will come to it later, but in general, to see possible effect of parasite immune interaction with data. Just limited data, I would say clinical data collected, basically patients, malaria, this is specific for malaria now. So what they've done, they've taken their isolates and cross-react them with serum antibodies to see the effect. And the picture that they look, what happened at the initial stage, acute infection, and 3 weeks later, when we're past this acute stage. So what happens, and again, without going too much into technical detail of this analysis, the idea was to see how strains in some way, although you don't see specific strains in here, but how they could possibly change in the course of this infection and identify certain patterns. Anyway, so that's what this work is about. So in some way, brief summary of these things, that initially, since it is a random sample of patients, right, with presumably carrying different strains of malaria, whatever they would be, more disordered states come to some kind of ordering. Whatever this ordering means. And of course, there is no any indication what specifically these strains are. It's pure empirical data. So in terms of modeling, of course, questions like this could be understood in terms of modeling. And there was a lot of work done in general about trying to understand parasite immune interactions and its effect on evolution. should go back. Okay. Okay. Sorry. Yeah. So different ideas were proposed. As I said, my motivation was to test some of these ideas. One I mentioned already, evolution of virulence by imperfect vaccine. And it wasn't only theoretical work or modeling work. What they did in Andrew Reed's lab, they sacrificed a great number of mice to test this idea. So to transmit it in some kind of serial passage experiment and to observe, are they become more virulent? Does it happen? Or in some version, if you like, not real vaccine, but some version to regulate it. Right. So another more theoretical, more theoretical approach to this. Now, these are more theoretical papers, modeling papers, really, to understand what goes under the name stress selection theory. And the question again that they ask, how again immunity can shape evolution of certain traits. What happens? Some of these models are population-based, some agent-based models. I will talk about it. Again, I won't go into detail. They've done this experimental work. Of course, they also try to propose modeling approaches to explain it. And ideas behind this modeling approaches was to understand the, if you like, interaction, trade-offs between traits, what they call phenotypic traits of this different strain: virulence, transmissibility, persistence. Okay, and there's a scheme again. I don't want to go too much detail, and many theoretically implications or conclusions that were drawn from these studies that typically what immunity does to diverse multi-strain system, it tries to drive them into distinct clusters, group. How distinct? Immunogenically disjunct, simply. The less cross immunity is there, the somehow more likely that this kind of picture that they will arise. So again, the motivation for my work was to test these ideas and see what else could happen with ABM. Right, and this agent-based model has some similarities, you will see, to what people, EMOT people do. Okay, so how it works? Just a few, just to remind you briefly how, basically to set it up like this, we have to know something about biology, basic biology of malaria parasite. And so it works like this. Whatever happens in humans is a sexual stage of this parasite. Sporozoites come in after mosquito bites. They invade red blood cells. After roughly 2-day cycle, 3-day cycle, they burst red blood cells and release more copies of this merozoites. Oh, sorry.
It's okay. You have to use the mic or people online can't.
Oh, sorry. Okay. A merozoite, this stage, invades red blood cell, develops into, basically divides into several copies. They burst red blood cells, and then the cycle continues. And once the cycle continues, it can continue for immunity starts to work. How exactly it works? Right. Key feature of malaria parasite, not only malaria, but malaria especially, is what it is evading immunity by means of antigenic variation. So, again, I will show how it works specifically in the context of malaria. By basically, what malaria does, it at each cycle like this, at each cycle, it can express different antigen on the surface of red blood cells, right? Called variable surface antigens. So as they are expressed, they stimulate the production of antibodies, specific antibodies. But on the next cycle, when next generation come in, some of them at least can switch this VAR gene to a different versions, and it allows it to evade host immunity, which has already developed. Okay, so basically, by looking at a typical history, parasitemia, say, in-host parasitemia, one observes these different ways. 1, 2, 3, 4, and one explanation, of course, what happens is switching this family of genes, VAR genes. Okay, so that all must be put in the model, and I will show you how exactly it will be done. Good, so the so-called VAR genes, there are other families of this antigens. Now, that's the way now the IBM, and I'm going to talk about, is set up. Each parasite, so we're talking about multi-strain. So each strain is made of a combination of these 4 genes, 1, 2, 3, 4, which are drawn from large natural repertoire, whatever number you have overall. These 4 genes, of course, you can have many number of them. Then what happens when you have several of them, different strains within host, and within host plays an important role. They cross-react with each other. How they cross-react, also quite clear. They share some of these VAR genes, and sharing VAR genes already has the effect that antibodies stimulated by one of them can affect the other one. So that's the key feature. The model, again, some features of model are similar to, let's say, systems like EMOD, agent-based models, right? Show again what it features. We have target cells, red blood cell populations. Right? We have infected red blood cells, of course. Then about this infected red blood cells, there's not one single population. They can express different VAR genes. Okay. On these different cycles. Okay, and then of course, the crucial thing for anything in terms of evolution is transmission, gametocyte stage. Yeah, I didn't mention it on my previous slide, but some of these merozoites at certain stage don't continue invading red blood cells, replicating, but turn into gametocytes. This gametocyte circulates within host, and they would be uptaken by next mosquito. That's how this transmission goes on. So that's what we try to account into our model. In terms of immunity, we'll have 2 levels of it. Basic, sorry. Okay. Yes. In terms of immunity, again, we have 2 innate immunity, which just basically a response to total parasite burden, febrile response. Usually that's what first happens when you get your malaria fever. And it suppresses somewhat this process, infection process. And then most important thing for selection, especially adaptive immunity. As these specific antibodies develop, of course, they then will selectively attack the specific clones which express a particular antigen. Good. So the model does account for all these processes: invasion, depletion, production, merozoite replication, antigenic variation, yeah, we'll show you, and gametocyte production, of course, very important. And on the immunity side, stimulation, clearing of targeted clone. The immunity can be lost, of course, it doesn't last forever. So all of this is included. Good. And transmission is also accounted for. We don't explicitly include, although mosquito, by the way, can be included. Indeed, it's an extension. Haven't been done, but can be done. And it is very important. Why? Since what happens within mosquito, now parasites can mate, and you have genetic crossover. They can swap their VAR genes, and you can generate much greater diversity in this part, in principle. Okay, so that's what the model includes. And just again, in illustrations, I won't go over all detail, but host, within host state, represented by big populations of the different cells, red blood cells, the infected cells, which again infected by different clones, this A, B, etc., indicate different clones. And for each clone, we also know at any particular time what specific variants they express. Okay. That's number one. Number two, then of course, gametocytes released by these different clones and immune effectors that they stimulate, specific antibodies. And this is again a schematic view. Again, what you see in the schematic view, let's say 4 different clones. Once again, they share certain VAR genes. And this diagram shows you how schematically they can interact with specific antibodies. Collection with specific antibodies. One thing to keep in mind, the genotype space for this multiclonal parasite, of course, is very large. Basically, how many? And in real life, of course, there are about a few dozens of these genes, 50 to 60. I'm not going to do simulations 50, 60. Probably is not also important, but at least this number of genes that you expect times what is the nature repertoire which exists. Okay, it could be a very large number. Of course, you have, in terms of immune effectors, there are far few of them. You have as many of them as you have your VAR genes in natural repertoire. Good. And the whole question is then how this whole thing works. That's what I'm going to discuss, and how evolution and selection works. And the model is run, to make it simple, on discrete time steps. Each time step is a cycle. One replication cycle, good, merozoite cycle. Okay, so some examples, simulations, simple simulations. One thing about this model, maybe I should still bring it for a minute. This one feature of the model, we don't have to prescribe in advance the number of clones are stored. They can change, evolve. Why? Since the model works like this, at a particular stage, a particular clone can be cleared, but you can get infected with a new one. And you can have unlimited number of these infections, so as long as I get. So in that sense, it's an open type system when you don't prescribe in advance the number of variables that you have. Good. There are just a few simple demonstrations of this model. I took 8 clone versions, right? This is it's a single clone, but it has 8 VAR genes, whatever ordering means. Oh, by the way, VAR genes, in my assumptions, other people with similar assumptions, the only difference, in fact, between different VAR genes, they have different replication number, okay? And then, let's say, ordered by this number of copies than they do when they invade red blood cells. Good. In this particular history, and this is history, and again, this is the number of replication cycles. So it would be something like 160 days history of this infection. So what you see here is depletion of red blood cells. Shaded part is how much. So again, you can develop severe anemia, of course, one consequence of malaria infection. Then next plot illustrate what happens to parasitemia. This is log scale, of course. So you see this multiple ways coming in, and the explanation is again straightforward. At some stage, they start with a particular, let's say number 1 variant, VAR gene, then they switch to number 2, 3, 4. And you see also clear what happens to immune effector, the specific antibodies as they evolve. Next thing, for all questions as far as evolution selection, what matters is not parasitemia, but gametocytes. Of course, you don't have many of them. It's after all one single clone. Gametocytes don't care about any expression. It's just gametocytes produced by this parasitemia. And that is this trajectory, if you like, path of gametocytemia. And next thing, and this is really crucial, whatever selection and other features will come in has to do with what is the probability of mosquito uptake this gametocyte. Well, what does it depend on? Well, it depends on simply how many you have of them per microliter of blood. Usually mosquito takes 1 microliter of blood. So we have sufficient number of them. We have the Sykes threshold. You can estimate this probability. And then from the history, you can compute simply this process. Again, it's very crucial. That's what I call TP, like in Andrew Reid formulation, not transmissibility, but that's simply transmission potential. So this is the crucial feature. And this is, so basically why it's significant. And it's not so much code itself, but cumulative effect. You can think of mosquito randomly biting a particular host like this. Of course, the larger transmission potential, the more likely it will get transmitted. Okay, so that's the crucial output. Oh, one thing I also mentioned here very briefly is that qualitatively at least, what model simulates is similar to real data. This is malaria therapy data collected long time ago from patients who were infected with malaria for whatever purposes of treating neurosyphilis. Qualitatively, good. So we move on. Yes, the phenotypes that you observe in such simulations, two really basic, one principal one, virulence in some sense, and you can measure it in different ways. It's all related to depletion of red blood cells. I mean, you can measure it, although as you will see, it's not essential feature of selection. And then really essential for selection, why since it all involves transmission, is this transmission potential, which is again cumulative probability to mosquito transmission. So good. One thing also about these phenotypes, they are not intrinsic, as you will see. We'll show you many examples. Why they depend on specific infections, specific mixture, for instance, of different clones within host. But the next maybe more interesting example, what you saw on the previous was one single clone. Now we mix them together, and this is very important since in terms of evolution and selection, really what plays essential role here is co-infections, when one host can carry several of them. If they live separately, that's somehow, well, it's unrealistic and has not really big implications. So these are now 4 clones. What is special about the system? They share only one VAR gene. In fact, it's the same one for all 4 of them. All others are distinct. So you run this now multi-clone infection for different clones. That's what happens to, again, red blood cells. That's what happened to parasitemia. But now each color represents not different variants. I'm not looking at variants, but each clone separately. And what happens is that the number 4 of them grows more faster, reaches peak suppressed, then other ones come in. And again, there's an interplay. They affect each other through these combinations of origin which are present. Good. So that's the question is, can you explain the outcome? Sorry. Also, let me tell you something about the outcomes that you see here. If you run these 4 clones separately as single clone infections, you get these values of transmission potential, something around 40, whatever units they are, right? Now, when you run them in this mixed infection, since they change, some of them lose somewhat this transmission potential. So again, the difference is between in this host, you have one with single clone run, and the other one, all 4 of them. Yes, that's the difference of transmission potential. But there are also big differences. For instance, number 3 is a big loser. Lost about half of the transmission potential, which number 4 gain. And the question, can you explain it just by looking at this cross-reaction network, right? How they cross-react through the, and even more detailed version of it, you wouldn't be able to predict it. There are some very subtle effects here. Why? What happens? That's number 4. So its combination was suppressed significantly, but not completely cleared, eliminated. Oh, by the way, the shaded here means detectable level. Anyway, it was suppressed, not completely eliminated, but because of this, it got a chance to rebound at a later stage, and overall, PP, in other words, number 4 gained by playing this game with other clones. So some of them lose, some of them gain. I cannot predict it from the makeup alone. Okay, so there's several takeout of this simple. I have many examples like this. Of course, that transmission potential is not intrinsic trait. Why it can change depending on particular infection, mixed combinations, right? Then mixed infection can enhance or decrease. And again, these are all essential features for immune selection. So in other words, you can say that sometimes, in some cases, they cooperate. In other cases, they compete, and you can't even say in advance what will happen. And that's more than saying that you just look at the genotype itself, combination of specific variations, you won't be and mixture, you won't be able to predict it. Good. So next thing, how to run evolution in such system. Now, big questions will come in. How would they evolve? And of course, they evolve on two levels. Number one, within host. So what am I going to do? I will show you. We run large ensemble of this host, naive host. It's something like young children, right? And we ask, and we'll subject them to different combinations of these strains. And when we ask, what will be selected? What of them? So this is one level of selection. And of course, more important than what happens on the level of communities. What can be selected? Then I will do, I will show today, more simple version of this community, what is called serial passage. We'll just track the evolution in particular line of host from one host to the next host. Okay, along the serial evaluation, right? So, and again, these are big questions. Which clones get selected? What role the virulences we'll see, but plays minor role, TT plays role, and then environment. Important things, of course, that we can experiment with. Subject this host, if you like, transmission lines, whatever, to different level of transmission. Yeah, okay. See what happens. Could have, of course, implications. I won't talk much about control implications. So here, the big question really, how to identify the successful clones and teams in mixed context. You can design many type of concepts. I won't go over detail. This particular whatever structure design allows you to do the following. You can track fitness, not only of individual clones, but teams, groups of them, couples, triplets, whatever. Model allows you to do it. And this is, again, very important for transmission. So some results, with demonstration, there are many more of them. For one thing that I will not spend much time at all, individual infections play a minor role in selection here. Everything is about mixed infections. And also, this virulence phenotype is not particularly significant drive factor. Now, mixed infections. So that's, again, what I've done. I've run already a large ensemble of these simulations for this ensemble, and a particular clone had chances to compete, if you like. Again, many other combinations of these clones, for instance, couples, triplets, and so forth. And then you ask, what are outcomes? Different TP outcomes, the distribution of these outcomes, so that what it shows you. And of course, there is number one feature that you observe. There is a loss of transmission potential fitness in mixtures, larger mixtures. You lose your transmission potential when you play this game in mixed infections. So it's one feature. Sorry, shows you here. Okay, sorry. One observation. You can also study on the level of distribution for the whole ensemble of simulations. Again, I skip the detail. Let me move a little bit faster. This is just illustrations. Another outcome of this statistical contest samples what happens, what it shows you, by the way, what happens when a particular clone is tested in couples, triplets, quadruples, and so forth, means again, infection with 2, 3, 4. What happens to fitness? Well, basically, again, you observe that's again the typical feature, loss of fitness. Somehow they overall lose. Okay, but interestingly enough, and this is something that was never mentioned in any of modeling studies, theoretical, in some cases, it can gain fitness. So a particular clone playing this game can gain fitness. In other words, it could be helped by others. So not only they compete through cross-reactive immunities and can cooperate. Okay, so this again, I'll take further to see how these ideas play. And this is about, it's not completely general community transmission, but a transmission in this serial passage line from recipient, from donor to recipient. So basically what we do, and by the way, all these hosts will be naive hosts in this simulate like children. So one child got infected, then mosquito uptaking gametocytes from first to second and so forth, and you follow, and again, they're all mixed infection, and you follow this chain evolution. And you want to know what happens, how they involve this. There's a special case of community transmission. One can, of course, run it more general, and this is some result. So we're on our many. So what this diagram shows you here, it's not time in any units of time, but rather how many transmission cycles we track. What you see here are different clones, a couple hundred of them or whatever. And then we follow what happens for these different clones in this transmission cycle. Now, on transmission cycle, of course, they get mixed. And they get mixed maybe with other clones. In fact, we even randomly add additional clone to this mixture to see what gets selected. And of course, what you see, some of them don't really last long, others run longer, but there are also really something which is very persistent. In other words, there are some clones which can last over many, many transmissions. So this is a selection. That's how a selection works. And the question that we ask, what are these clones? What is special about them? Why some of them manage to do it as opposed to others? And the idea is, I won't go into too many details, but the way they cooperate, again, it's not only, you see, in simple strain theory, theoretical models, they should expel each other. They should somehow partition into disjunct clusters. They don't do it. In some way, they organize themselves. It's a different level of organization. In some way, partly cooperate in cliques. And they can even share some other members within these cliques. That's how they are able to succeed. So one interesting feature of this model, simulating this model, this new feature, the effect that immunity can not only split them apart, but also help them to collaborate and get selected. Okay, so just summary, there's still preliminary results. More work has to be done. And of course, this can be extended. It just wasn't done yet, but it's doable, of course, all to more realistic ABM communities now with proper features, other features. You can distribute this host somewhere. You can allow genetic crossover of these different mixed infections in mosquito. You can study. Next, questions, how transmission intensity can affect selection outcome in systems, okay? Just changing transmission intensity has some implications. I didn't show you, but anyway. What is the makeup of this cooperative teams, cluster, whatever you want to hold? So again, selection doesn't happen on simply individual level. One particular clone is not a super clone. In fact, right? They work in teams, and teams in some way can maintain clonal diversity in host communities. These are all questions that can be explored in principle. More resilient, they could be, again, I'll speculate here, I haven't done it yet. But to test, are they more resilient to interventions? Like transmission reduction, drug, and so forth. How do they respond and adapt? And the next question, if this indeed holds, what implications it will have for control? Thank you.